It is my pleasure to introduce with today's speakers, Dr. Ayman Oweida. I had the pleasure to meet him since he was studying his PhD, and I started working here. So he obtained his PhD in experimental medicine from the McGill University. So he is a McGill alumni, and his PhD was specializing in radiation oncology under the supervision of Dr. Bassam Abdul Karim. And his postdoctoral work was at the University of Colorado, and he focused on understanding the effects of radiation on tumor immune responses in head and neck cancer. He's currently an associate professor in the Department of Medical Imaging and Radiation Sciences at the University of Sherbrooke, and he holds the Canada Research Chair in Antitumor Immunity by Radiotherapy since 2020. His recent work focuses on the role of immunosuppressive cell populations in response of solid tumors to radiotherapy, and he also works on developing non-invasive imaging tools to track immune cells in tumors. So he's actually doing a lot of work that is very hot topic in radiation oncology nowadays, the responses of the immune cells and the role of immune cells in antitumor and pro-tumor responses. So help me welcome him today with us, and about his research.
Thank you very much, Norma, for the kind introduction. I'd also like to thank the Medical Physics Unit for this kind invitation. Really, really happy to be here. Actually, first time I walk through these halls since I graduated in 2015. So I had some ecstatic feelings coming here. And I'm really happy to also be able to share this research and maybe discuss any questions you might have, and opportunity to collaborate in the future. As was mentioned, my topic of interest in general, and I'll talk about the different streams in my lab, is on overcoming immune-mediated resistance in solid tumors. So I thought I'd start by, oh yeah, the mouse is not my mouse. Thought I would start by dissecting the title a little bit. And one of the key words in this title is radioresistance. And the definition has not really changed much for many years. In the early 80s, radioresistance was defined, a tumor was defined as clinically radioresistant if it regrows within the irradiated region, regardless of its rate of regression. So that last phrase was important because when tumors regress rapidly, it was thought that they're radiosensitive if they're dying, at a certain rate really rapidly. But then what we found is that they could be dying rapidly, but then there's clones of cells that are selected that remain dormant, and then the tumor comes back. And so that was a definition of radioresistance that really has not much changed since then. In 2020, a definition from a review paper, it says tumors are labeled as radiation resistant if recurrences are observed within 6 months following the course of radiation. And that's because we needed to distinguish between what's a recurring tumor due to radiation and therefore radioresistant, and a new tumor. And also the causes of clinical radioresistance, believe it or not, like the definitions of these pillars of radioresistance have not also changed much in their existence. So there are 4 related factors that are mentioned in this 82 paper. Tumor-related intrinsic factors could be DNA damage, and we'll talk about that in a bit. Host-related factors, technical factors, and probabilistic radioresistance, so meaning clones that are selected for radioresistance. Interesting thing was, at the time, so host defenses, there was a clause here that says attempts to exploit specific or non-specific immunologically based host defenses in the treatment of human cancer have been almost uniformly unrewarding. So that's probably the only thing that has changed, because it has been very rewarding to exploit immune-related radioresistance for improving cancer treatment. So for an audience like this, I don't think this is new to you. Radioresistance, you have intrinsic cellular mechanisms related to the tumor's ability to repair DNA, to modify the cell cycle, so downregulate pro-apoptotic proteins, and to have elevated antioxidants that suppress oxidative factors. And then you have the tumor microenvironment, and one of the ones that has been exhaustively studied is hypoxia, cancer stem cells, which continues to be an active area of research, and then the immune component related to the tumor microenvironment and how that can mediate radioresistance, which is my focus of this presentation. and what I think is a really hot field that has a lot of potential and promise to improve radiotherapy treatment. So what do we mean when I say immune-mediated radioresistance? So, radioresistance, we can a little bit appreciate that, but immune-mediated radioresistance. So the best way to define that is by talking about immune-mediated response to radiation. So what do we mean by an immune-mediated response to radiotherapy? One of the classic studies that has been used to demonstrate this was to use, and so this is work from Ralph Voxelbaum, which I highly encourage anyone interested in this to read his work. He continues to publish really great work from the University of Chicago on these topics. But he did a study where they compared implanting a tumor in a wild-type mouse or the equivalent mouse that has no T cells in it, so a T cell deficient mouse. And then they irradiated the mice with 25 gray in one fraction, and they followed tumor growth. And what you find is in mice that are T cell deficient, you could say these are radioresistant. There's no response to radiotherapy, 25 gray, one fraction, tiny pea-sized tumor. And then in the wild-type mice, you find that there's a significant decrease in tumor growth. So their conclusion from this simple experiment was that T cells were mediating the response to radiation. To further prove that, they took now the same mouse, forget the T cell deficient mouse. They took the wild-type mouse, and then they depleted what they thought was the cell that's causing radiosensitivity, and that's the CD8 T cell, which oftentimes is responsible for killing cancer cells. And now they gave a higher dose because they wanted to see if a higher dose of radiation could further improve response to radiotherapy. So you can see with 45 gray and 3 fractions, they were able to obtain a flat line almost. If they depleted the CD8 T cells in that same mouse, they reversed the effect. So there's no longer a response to radiotherapy in that same mouse if you deplete the CD8 T cells, indicating that you have an immune-mediated response to radiation. If the CD8 T cells are there, you have response. They're not there, you have no response. Another example to talk about immune-mediated response to radiotherapy, as a lot of you have heard, is through the abscopal effect. So first defined or shown by Robert Moll in case studies, and then ever since then, we see once in a while these rare case reports of abscopal effects. In mice, we've been able to actually reproduce this way more than we have in humans. And so with the abscopal effect, what we see is that radiation can initiate an immunogenic signal, so in the primary tumor, that causes systemic activation of immune response against tumor cells. So this is a beautiful case, 2018. You irradiate 60 gray breast tumor, you irradiate the colon with 39 gray, you end up with complete elimination of all disease in this patient. That was not the intended goal, of course. It was a palliative case. And then you end up with a rare case of an abscopal effect here that's mediated by the immune response. So in some of my postdoc work, I went a little further to dig into mechanisms of how do immune-mediated responses to radiation occur. So we found that, for example, if you take cancer cells, these are head and neck cancer cells, and you irradiate them with increasing doses of radiation, you find that you upregulate major histocompatibility complex 1. So it's the way cells present antigens. So if you upregulate this protein, then that means these cancer cells are presenting antigens way more to immune cells. So that's one way, and CD80 is a similar molecule, and then programmed death ligand 1 also is upregulated. And then if you look at T cell proliferation after exposure to irradiated cancer cells, you see that T cell proliferation and activation are increased after they've been exposed to irradiated cancer cells. And so to conclude a little bit of this, radiation can augment antigen-specific anti-tumor immune responses by increasing the activation and proliferation of T cells, altering chemokines that preferentially recruit cytotoxic T cells and upregulate MHC1, and promotes the release of tumor neoantigens and activation and migration of dendritic cells. Now we can talk about immune-mediated radioresistance. So you saw how immune cells could be the mechanism by which radiation works. Now, what do we mean by immune-mediated radioresistance, which is really the topic of my research? And we can say that it's resistance to radiotherapy that is driven by immune mechanisms, such as the recruitment, activation, and sustaining of immune cells, as you'll see some here, that promote tumor growth. So there's, for example, these 3-cell population, regulatory T cells, myeloid-derived suppressor cells, and macrophages, tumor-associated macrophages. If a tumor successfully recruits these to the site, then this tumor could potentially be using an immune-mediated mechanism of radioresistance. Similarly, if a tumor is able to induce the expression of immune checkpoints, such as PD-L1, which I think we've all heard about, then that tumor could potentially be also turning off the immune system and therefore having an immune-mediated resistance to radiotherapy. So one piece of data that I wanted to show here is related to, this clause, when radiotherapy works, immune cells are at the forefront. And you saw this in Wexelbaum's work when I showed you that CD8 T cells, when they're there, then radiation works. When you take them out, radiation doesn't work. Well, similarly, when radiation fails, we can find also another immune cell type that can be the culprit. So I'll talk about this in more detail later, but for now, in this work that I showed earlier, in my postdoc, we looked at regulatory T cells. And what we found was if you irradiate these mice with head and neck tumors in the buccal cavity right here with 20 gray of radiation, you get a small response to radiation. If you deplete the regulatory T cells, which sustain tumor growth, then you get 4 out of 6 mice eradicated tumor. So complete eradication of the tumor if you deplete these regulatory T cells, which I'll talk about, like I said, in a bit. So this is all to demonstrate or to define what immune-mediated radioresistance looks like. And so with that little introduction, I wanted to now maybe go a little deeper into some of our own results from my lab at the University of Sherbrooke. So this is pretty much my attempt at dissecting the title that I gave you for this talk. Mechanisms of immune-mediated radiotherapy resistance is the work that we do. I have 3 streams in the lab. One driven mainly by Cleo, a PhD student in the lab and a recent MSc graduate, on the study of the immune tumor microenvironment in response to radiotherapy. Another stream that was led by an MD student and a PhD student, Rima, on combining therapies involving radiation and immunotherapy. And a third stream that was led by a recent graduate, also Noor, on imaging methods for non-invasive immune phenotyping of tumors, where we use PET and radiolabel isotopes, radiolabel antibodies to track T cells. I'm going to mainly talk about this one, but I'll also, a little bit at the end, address both of these research topics. So to begin with, we needed a clinical problem. We needed to address a place where radioresistance has a clinically unmet need. And in head and neck cancer that's non-virally driven, so HPV negative head and neck cancer, local regional recurrence, hence radioresistance, remains a problem. So 30% of head and neck radiotherapy patients develop local regional recurrence after curative RT. This is an example from Dr. Shangshu Wang at our hospital, showing a patient who had 70 gray and 33 fractions to the right oropharynx and lymph nodes. 3 months later, they had no residual disease. And 6 months later, the tumor was coming back within the irradiated field. So we had recurrence here. And when recurrence happens, it's even harder to treat, whether it be with salvage surgery or re-irradiation. So we created a translational animal model for this kind of cancer, which like I said, we needed a clinically unmet need, and head and neck cancer radioresistance was a problem. So we have these orthotopic animal models where we implant tumors in the buccal cavity. So we inject the cancer cells, inside the gums of the mouth, and you end up with a head and neck tumor that looks like this, with draining lymph nodes that can easily metastasize to, similar to what we see in patients. If you irradiate them, you don't get much of a response. Now, if you irradiate them with a higher dose, 8 gray times 3, 24 gray, 24 gray total dose, and add anti-PD-L1 therapy, you still have tumor progression despite the combination of radiotherapy and immunotherapy. And that's the red line here. And then blue is 24 gray on its own. And then green is 10 gray plus anti-PD-L1, and purple is 10 gray alone. So in this experiment, we asked, okay, it seems we have a radiosensitive or a sensitive phase right here and a resistance phase right here. So if we acquire tumors at 1 week after treatment and another set of tumors at 3 weeks post-treatment, what kind of immune markers are we going to see change over the course of time? And so what we saw looking at the purple here for a second is TIM3, another immune checkpoint that we thought maybe tumor cells could be using to shut off the immune system. And we're going to and I'll show you how we targeted that. But you also see that anti-PD-L1, which we thought didn't work well here, is generally low. So the red is anti-PD-L1, goes up a little bit with radiation, but it's generally low compared to TIM3. When you block, so I'll just take you to the end of this. When you block TIM3 plus anti-PD-L1 plus radiotherapy, 24 gray in 3 fractions, you still have tumor progression. So highly radioresistant, and we haven't been successful at using an immune marker or an immune mechanism to improve treatment for this kind of head and neck tumor. Finally, we said, why haven't we looked at the green? The green shows FOXP3, which is the regulatory T cells. FOXP3 is a marker of regulatory T cells. And to do that, well, before that, we looked at the Tregs, the cells themselves, and we found that in the early phase, they're 15%. After, in the late phase, it goes all the way up to 60%. So we thought maybe this is the culprit. And this takes us back to the graph that I showed you earlier. When we depleted the Tregs, we found that we had 4 out of the 7 mice cured. And so it seemed like these were the driving mechanism for radioresistance in this tumor model. And not only was it 4 out of 7, the 3 that remained, that progressed, we went and analyzed them, and we found that they still had Tregs, which means we had a problem depleting their Tregs in those 3 mice. In the other 4 mice, their Tregs were completely eradicated. So depletion of Tregs restored sensitivity to radiotherapy in this case, and it was not anti-PD-L1, it was not TIM3. All to say that we need a personalized approach, of course, to figure out what mechanism each patient has. So my student went on to ask a few questions. One, how do these Tregs come to the tumor? How did they make it there in the first place? And does radiation promote the activation and proliferation of these Tregs and migration? So for the first part, we looked at all of these cell lines that you see here in a public data set, human cell lines, and we put all of these chemokines. Chemokines are factors that the cell secretes as a signal to bring in immune cells. And we analyzed these chemokines, which are all involved in Treg migration. And we found that CCL20, which you see here, is the most common chemokine expressed by head and neck cancer cells. In mouse cell lines, these are all human. In mouse, we had 2 cell lines, and we had one MOC2 that had high expression of CCL20 and another that had low expression. So all of our further studies are now on the MOC2 cell line because we thought this is the one that could be recruiting the Tregs. Then we asked, does radiation promote Treg migration? So we co-cultured Tregs on the top, cancer cells on the bottom. And we irradiated these cancer cells or did not irradiate them. And what we found was if you irradiated cancer cells right here in red and then cultured them with Tregs, you find that these Tregs are going to migrate more effectively towards the cancer cells. And if you block anti-CCL20, which is the green right here, so you irradiate and you block CCL20 at the same time, you reverse this increased migration of Tregs. So we thought, maybe radiation is also contributing to an increase in the Treg migration. And if we block it, then we can get reversal of this effect. We also checked for the receptor of CCL20 on Tregs, and we found that it is highly expressed on Tregs that migrate, but not the ones that didn't, which just confirms that there's a receptor and ligand coming together to work. So preclinically, we use the small animal image-guided irradiator to irradiate. You can see here, the buccal tumor. It's quite a big tumor. irradiated with Tingray, and we blocked anti-CCL20 with the hope of seeing that similar to what you saw with the CD25 when you deplete Tregs, but now we're not depleting Tregs, we're actually blocking them from coming to the tumor. And so this just shows that indeed it works. When you block anti-CCL20, you reduce the proportion of Tregs in the tumor as shown by flow cytometry, and you increase the CD8 T cells, which are the good cells that we want. And if you look at tumor growth over time, although it's a short period of time, you find there's a significant reduction in tumor growth, but it's not impressive. This was not really, something that excited us. And my next student then went on to the drawing board and said, if we follow these tumors further after day 15, are they going to continue to grow? And that's what she saw. If you have radiation alone versus if you have radiation with anti-CCL20, tumors continue to grow uninhibited. So we went back to the drawing board and we asked what other immune cell populations could be involved in this tumor. And we found the green here, the M2 macrophages are highly abundant in the MOC2 tumor model. And then we asked which chemokine, remember those are the secreted factors that bring in immune cells, could be responsible for promoting the migration of macrophages this time. So now we switched gears to look at the macrophages and whether they could be the culprit. And you can see here that CCL2 was really high, and CCL2 is a major driver of macrophage migration. So we thought maybe what's happening here is we have both the tumor-associated macrophage and the regulatory T cell. Both of them are inhibiting effector T cells and antigen-presenting cells like dendritic cells. So we further pursued macrophages, but eventually we want to block both. I don't have data on that yet, but eventually we want to block both populations. So we saw that in the MOC2 model, which remember is the highly radioresistant model, radiation increases CCL2 expression. In human cancer cell lines, we saw that in one of them we have that, in another we don't. So it's always nice to have a positive and a negative. And then to confirm at the protein level, we found that indeed CCL2 is increased in response to radiation. And to look at the secreted factor, we found that radiation also promotes the secretion of CCL2. So now we did a similar study as to the one you saw earlier, where we co-cultured macrophages this time with irradiated or non-irradiated cancer cells. And what we saw was so this is macrophages alone. This is their migration on the chamber. So we just take the chamber and we stain it for the nucleus, crystal violet. This is if you add non-irradiated MOC2 cells. So these are what you see here are macrophages that migrated upon seeing cancer cells. So cancer cells are secreting cues to tell these cancer cells to migrate towards it. Imagine this case now in the tumor in the body, when the cancer cells are secreting the same chemokine. Now you put irradiated cancer cells, and this is what you get. So you have a significant increase in M2 macrophages. And if you block CCL2, you completely reverse this effect. And this is shown quantitatively over here. Then we went in vivo, and we said, if we look at irradiated mice, do we really see an increase in macrophages? So then we stained for CD163. And this is an example from a section that was non-irradiated, and this is an irradiated section. And we see that macrophages, and in particular, they're called M2 macrophages, are significantly increased and abundant in the tumor after radiation. So finally, before the preclinical study that I'm going to show, we asked, does this chemokine called CCL2, which I just said promotes migration of cells? The problem with what I just said is we don't usually see in the circulation M2 macrophages. What we see is monocytes. So how is it that these M2 macrophages are getting to the tumor? Are they coming in as monocytes, or are they really going into the circulation as M2 macrophages? So we said, does CCL2 promote M2-like polarization. So essentially monocytes coming and then getting polarized to M2. The reason we asked also this question, I mean, it's because it's an important question, but also there's a paper that showed that CCL2 can promote macrophage-associated chemoresistance, so not in the context of radiation, via a particular mechanism shown here I won't talk about, but essentially they showed that iNOS, which is for M1 macrophages, and M2, which is for M2 macrophages, M2 is increased and iNOS is decreased when combined with recombinant CCL2. So it's a valid question to ask, and there is a study that's been shown that shows that. And so the student, Cleo, did an experiment where we have an experimental arm. We take monocytes, which are the ones that we see in the circulation, and we co-culture them with CCL2, the protein, recombinant form that we just order. And then we see whether they can become M1 macrophages or nothing, they stay as they are, or whether they will polarize to M2s. And in the control arm, we have the monocytes, and then we polarize them using established protocols to M1 or M2. So what you see here is interferon gamma LPS versus TGF-beta, IL-4, IL-10, which is the classic way to polarize them. So this is M2s polarized using the classic method with TGF-beta, IL-4, IL-10. And this is when they're combined with recombinant CCL2 instead of all of these. And you can see how CCL2 does have the capability to polarize monocytes into M2 macrophages, not so much to M0 or M1, which is the orange. So finally, we did the preclinical study. After all those results were since they were reassuring. And in this case, we irradiated twice, 10 gray on day 8, 10 gray on day 10, and administered anti-CCL2 to block the migration of these monocytes and inhibit their polarization based on the data that we just showed. And this was really encouraging. The data was actually quite promising, and 2 mice continued to have tumor shrinkage until the end. Data from the blood, so the plasma of these mice, showed us that indeed the inhibitor is working. So there is no CCL2 in the plasma. There's a significant reduction in CCL2 levels. And then we saw that the M2s are decreasing at the expense of the M1s. So M1s are the good ones. So there's a significant decrease in the M2 macrophages. When you're using anti-CCL2 in combination with radiotherapy, you have a significant decrease in tumor growth. Finally, I have this recent clinical study that we started where we are collecting patients' blood samples from patients who are being treated with radiotherapy. So for the most part, head and neck cancer patients, and some who are not treated with radiotherapy. So it's a collaboration between a surgeon and a radiation oncologist. So we collect blood samples from head and neck patients who don't receive RT and others who do. And what you see here, we're looking at CCL2 levels in the plasma. In patients who don't receive RT, we follow them over 6 months, and we see no significant change. In patients who received RT, there's a significant increase in CCL2. Of course, there's a need to look at individual patients. This is looking at group population changes. We want to be able to do additional analyses and correlate for individual patients the CCL2 levels with outcomes, and also with intratumoral M2 macrophages if we can. So to summarize this project, chemokines, I talked about CCL2 and CCL20. Sorry for the confusion. They happen to be coincidentally 2 and 2, but there are well over 60 chemokines that have been identified. So there could be others at play. But the idea is that chemokines that are secreted by cancer cells, whether at baseline or that are induced by radiation, they could dictate the immune tumor microenvironment phenotype and whether a tumor will respond or will not respond to radiation. Tumors infiltrated with Tregs and M2 macrophages I didn't talk about myeloid-derived suppressor cells, but they also play a role. Tumors that are infiltrated with these cell populations tend to be highly radioresistant. And so we need additional treatments to mitigate the negative effects of these tumors that are immunosuppressed. And a holistic approach to targeting these immunosuppressive cells or their chemokines is necessary. And I say this because, what if a tumor has all three of these? What are you going to do then? You're going to target each of these three populations plus RT, that's three different drugs. And the answer should be that we need to find a way to target multiple cell populations using the same drug. So, for example, COX-2 inhibitors can target multiple immune cell populations. That's an area that we're thinking about in terms of drug repurposing. And so having an approach that can target multiple immune populations at the same time will be an important future direction. Another project that I told you about earlier is the use of radiosensitizers against the tumor microenvironment. And so for that, something like immune-targeted inhibitors. There's other, approaches that are currently being researched, such as, and this one's quite attractive, cancer-associated fibroblast-targeted inhibitors and other stroma-targeted inhibitors. In my lab, we're mainly focused on using, immune-targeted inhibitors such as anti-PD-1, PD-L1. And so Leah, one student in the lab, was looking at comparing hypofractionated versus single-dose SBRT in lung tumors based off of a couple of clinical studies that have compared these two dose regimens, the RTOG0915 and another multicentric study where they compared 34 gray in one fraction versus 48 gray in 4 fractions. So those are clinical studies. And so we wanted to do the same. We wanted to, but more from an immune perspective, to understand what happens from an immunological standpoint. We were surprised by the results at the subcutaneous level. So these are subcutaneous tumors, irradiated, sorry, I said 34, I should have said 30, and 48 gray in 4. And we were surprised that we don't see what the clinical studies are seeing. We see a huge difference between 30 versus 48 and 4. So 30 gray in 1, tumors continue to grow, they respond well, whereas 48 and 4, we're able to see tumor eradication in a lot of the mice. Both of these studies demonstrated that there's no significant difference between these two regimens. And then during COVID in our institute, at least, patients were allowed to get 30 gray in 1, and that's when they performed also a pilot study showing that both regimens work similarly in patients. What we found was at the immunological level, 48 and 4 is able to promote both an increase in CD4 T cells and CD8 T cells, but not so much the 30 gray in 1. Now she developed an orthotopic model. So we're going orthotopic soon for these studies. And we're going to look at the lung microenvironment after both of these tumor regimens and how they can be combined with anti-PD-1, PD-L1, which is pretty common in lung cancer. So I switched gears here to lung cancer. And so this is the next study that we're working on. She's irradiating 48 gray in 4 versus 30 gray in 1 and combining with anti-PD-1, PD-L1, and then looking at the lungs in terms of the tumor microenvironment and tumor growth as well. Finally, the last project that I'll talk briefly about is imaging the inflammatory response to radiation. And this is work that we're doing in collaboration with Brigitte Guirin at the Imaging Center, who's involved in radiochemistry work. And so the idea is to image CD8 T cells or T cells in general in patients, but we're going to start with our own mouse studies. This is not our paper. This is, I should have put the authors, but this is a paper that showed the whole CD8 T cell visualization before and during immunotherapy. So this is not radiotherapy, this is immunotherapy. And they're showing that the spleen, which is pitch black here, it's an immune organ, and all the immune all the lymph nodes actually light up because they have T cells. in addition to the tumor, which in this case was breast, I believe, and liver. So there's parts of the liver that light up. Regardless of the 3D part here, looking at this was a really nice image where they showed that using zirconium-89 radio-labeled CD3 and CD8 antibody, they show that there is indeed CD8 T cells in these patients. So this is in a patient population. And it even shows similar patterns where the high intensity correlates with square 1 here, which is the one that has the highest proportion of CD8 T cells. Number 5, for example, is the second highest, and you see that here as well. Number 4 is the least, and you see that here as well. Really consistent signals with the IHC staining that they're showing here. So we started this project by radiolabeling CD3, which is a marker of T cells, with a chelator for HMSA and zirconium-89. And we just started with basic studies showing that if you block this, then you get a reversal of the effect. You have competition assays. And then we looked at the biodistribution in the organs of CD3 radio-labeled zirconium-89. And you can see here all the organs, and spleen is the highest. It's an immune organ, so that's expected. That's where the highest proportion of T cells is. And then in the tumor, what we found was in the irradiated tumors, so this is in mice, there is an increase in the signal on average. So this is the tumor to blood ratio, for example, in 0 gray versus 10 gray, it's nearly doubled. And so this was promising work to continue to the pilot study where we looked at MOC2 tumors. So these are MOC2 tumors that we usually plant orthotopically in the buccal cavity. But for the purpose of this study, we wanted to do a flank model. And you can see here, this is zirconium-89 radio-labeled CD3 antibody. And there's even a ring pattern. We still need to do the IHC to confirm whether indeed there's a ring of CD3 T cells, for example, surrounding this tumor. And then we looked at colorectal tumors, but also sub-Q in the flank. And you can see here non-irradiated. So this is 0 gray. And this is 0 gray blocked. And then this is irradiated. And you can see the signal increase. And this is another irradiated. So to conclude on mechanisms of immune-mediated radioresistance, we need tools to detect and track immune components of the TME. Radiotherapy and immunotherapy combinations need further optimization. I showed you data regarding 30 gray versus 48 and 4, and how potentially we need to better understand these dosing regimens that are used in the clinic and how they work at the immunological level. And then cues such as chemokines from cancer cells need to be better deciphered to understand the crosstalk between the immune microenvironment and the tumor. So for the future, I just put these 3 boxes. We need tools to detect and track immune components. We need radiotherapy and immunotherapy combinations that are optimized in terms of dose, fractionation, and sequencing. Same question we've been asking for so many years, by the way. And we need to interrogate cues from cancer cells, so understand what chemokines they're signaling, they're sending out in terms of their communication with the immune cells. So with that, I think I'll keep it at that. And I'd like to acknowledge a lot of the students who worked in my lab over the years, collaborators, and sources of funding. And thank you again for the invitation. Happy to take questions.
Thank you very much for the very interesting presentation. I would like to go with questions from the audience here, and then I'll check if there's any other questions on Zoom. Any questions?
Kind of a general question. So I think this is really interesting, but then it gets me thinking, how does radiotherapy work at all?
Right. I agree.
What percentage, I guess, of the radiotherapy response is really immune related, and what percentage is not? what we traditionally think of.
Yeah, well, I think they're connected actually, and although that's hard to sometimes wrap our heads around it, but from a biological standpoint, these cancer cells that are dying, we also, I didn't talk about this, we also have something called immunogenic cell death. Could be, so they're dying via DNA damage, the classic way of how we think about radiation working. So whether it's the free radicals or direct DNA damage, indirect DNA damage, the classic picture in our minds, I think, is working there, of course, causing apoptosis and immunogenic cell death that eventually leads to the activation of immune cells, which then come, the activation, yeah, so the immune cells come and clean the mess. So you have, just like any infection, for example, immune cells coming to clean the mess, but they also recognize the antigens that have now been exposed by the cancer cells that died. And so what they do, the immune cells, is they contribute to further killing additional cancer cells that maybe the radiation did not. So essentially, the way we see it is they work hand in hand. There's some damage occurs by radiation and a continued damage that occurs by the immune cells. How do you decipher these two components and better analyze them? I think that's a really good question.
Yeah, from what if I just can add something, also that what they are mentioning with all these immune-mediated effects is that it depends on the dose of radiation. Apparently, below certain 6 to 8 grays, it is more like a pro-tumor response that we will get, that the immune cells are like the Tregs are going to be upregulated, let's say, and versus if we use a higher dose of radiation that will promote this immunogenic cell death, like above 8 grays, that's when we get anti-tumor immune response. So that's what I have read in the literature, and that's what some of the people working in this immuno-oncology, radiation oncology, are mentioning about the response. So yes, there's the different mechanisms that are involved in.
Yeah.
Maybe just a follow-up, but not exactly related question. We hear a lot these days about radiation-induced lymphopenia, which obviously
is the opposite,
right.
The effect. Can't compete with the effect, I guess.
Yeah.
You didn't mention it at all in your.
No, I didn't, but it is a problem, and I'm gonna let Bassem comment on that because I think
nowadays it looks like they are moving forward to not irradiate the lymph nodes, or at least not the regional lymph nodes.
So that's a big question.
This is a good question for the clinician here.
My question for you would about the group of mass that we showed that Treg contribute to the resistance. Of this early 5 weeks. Have you tested? I mean, you said you did depletion of Treg. You didn't mention how you did that. Did you inject like some?
No, anti-CD20.
Like an antibody to disease.
Antibody, yes. This antibody, anti-CD25 antibody directly targets it. Problem is.
For radiation or which is?
Great question. So actually from day 0. So we wanted to make sure that this tumor that develops in the mouse has no shot at getting Tregs in it. So there would be, 0 Tregs in the tumor from day 0. When you inject cancer cells, you have anti-CD25 antibody. So any cues coming from the cancer cells that are bringing the Tregs get depleted every day. You know what I mean? So that by the time we irradiate the tumor, we have a Treg-free tumor versus the original tumor, which is Treg enriched. So which is clinically impossible to apply because this is from the day of tumor inoculation. And actually, it's a big problem getting anti-CD25 antibodies to penetrate into the tumor clinically, like in.
So, question here that I have, you start early, but despite starting on the first day when you inject your cell, you still have a recurrent tumor. So in this recurrent tumor, did you have a Treg or?
Yes, in the 3 mice.
Yeah.
In those 3, yes. Yes, I don't know why. So ideally one would inject 10 mice with depletion antibody and check each of them that they attained depletion. Because for whatever reason, there could be technical issues injecting the mouse, or I don't know, like how the student that day was feeling, or So ideally you would check. We didn't. We only checked at the end of the experiment what proportion of Tregs were there in the circulation. And that's what we found, that in the 3 mice that recurred, their Tregs came back.
So you did not.
We didn't check
you did
during.
not check your
Yes,
Tregs.
exactly. Yeah.
I have a question.
Yeah.
So from the models that you're using, the MOC1 and the MOC2, have you seen the metabolic profile of those cells?
No.
Are they more glycolytic? Are they
Right.
less glycolytic? Because I'm interested in the
Yes.
metabolomics and how the metabolic profile of the tumors
Right.
play a role in the tumor
I'm completely
microenvironment.
naive to that. Happy to share and we can talk about it. But yeah, I haven't looked at metabolomics. I know a colleague who's looked at cancer-associated fibroblasts in these tumors and is targeting FAP, fibroblast-associated protein.
Seems like it's a lot like glycolytic, like very highly glycolytic tumors will have more lactic acid,
Yeah.
more acidic pH,
Right.
and that will trigger, like it will promote more Tregs, more M2
Right.
macrophages. So the
It's
pH,
true.
the acidity, and the tumor microenvironment will also be an expression of chemokines should
Yeah.
be also mediated through these pathways.
Absolutely. Yeah.
We can talk later.
Yeah.
And just one more
Yeah.
question. Just maybe you can just highlight a bit the work done on the comparing single fraction versus full fraction therapy.
Yeah.
You said your student has done the work on subacute, and now you're moving off.
Yes.
And you mentioned there is differential immune response.
Yes.
So do you expect this difference
We'll stay.
stay or even do well, but I want to add something to highlight that we have some clinical data
Yeah.
in early stage lung cancer combining SBRT with immunotherapy. Show for trial there will be no benefit.
Really?
Yeah, the study has been stopped because
Yeah.
on the interim analysis that when we combine pembro with a key fraction or 5 fraction, there was no benefit in the study is get it.
Yeah.
Yet. So we have opened the study with 5 or 6 patients. That is extremely disappointing that early stage they don't have this benefit from that's so important. Can you observe what are the difference in terms of the immune response we had between 1 fraction and 3 fraction or 4 fraction?
So the sub-Q model actually is consistent with what you just said. In the sub-Q model, I didn't show it here, but she combined anti-PD-L1 with 30 gray versus 48 and 4 and found no benefit for immunotherapy when added to radiotherapy. It was the same, actually. There was no difference. Whether it was the 30 gray in 1 or the 48 and 4 in the sub-Q model.
Yeah.
Now we want to shift all of this orthotopically because it's always question of whether it's the right TME and clinical relevance is not really attained with sub-Q models. So now we have the orthotopic, but I would expect what you just said, and I would expect that we need to go back to the drawing board and understand what is the immune mechanism in our tumor model. And then for patients, I don't know how we're going to personalize this one day, but each tumor is different. And maybe you need TIM3, CTLA4, I don't know.
But in your radiation treatment, just forget
Yeah.
the image. When you have a single fraction or 4 fraction, what are the differences in immune response that you stimulate with your treatment?
So we saw that the 48 and 4 was better. 48 and 4.
What the
CD8 T cells went up. Both activated CD8 T cells and CD4 T cells went up. Exhausted T cells went down in the 48 and 4. But combine it with immunotherapy, you get no additional benefit. So the immune surrogate of response didn't really.
But with a single fraction very great, without any stimulation or infiltration with CD4.
A little bit. It was not a lot.
Maybe everything is killed, so there's no cells like 8%.
It's possible. Yes.
It's the high dose. So what I've heard in some conferences is that when you use a very high dose, you kill all what's in there. So there's no opportunity for example, immune cells to phagocyte cancer cells to send the antigen to the lymphocytes T and then mount an immune response again. So that's why they are talking about a sweet spot for the dose of radiation. Too high is bad, too low is bad. In terms of too low, it's going to kind of improve like pro-tumor immunity. So it's going to help the tumors continue to grow. And then when you give a too high dose, you kill everything and there's no response or not immunogenic cell there, that's where the immune cells could present the antigens to the immune system and then attack the cancer cells. So that's why they are talking about the like doses of fractionation between 6 to 8 grays. At least that's what I heard the last time I went to the conferences. But this is always changing. Like you're showing a little bit higher doses, 12 grays working well for you. So you're getting like good immune response in terms of CD8.
Yeah, so this is like the CD8 T cells with the 48 and 4 and 30 and 1. And actually, so CD4 T cells, significant increase with the 48 and 4 and nothing with the 30 and 1. And then with CD8 T cells, they go up with both, but more with the 48 and 4. I think we, prematurely ended the sub-Q study, like to switch to orthotopic because a lot of questions remained about the validity of the model, but additional markers are needed, like interferon gamma, granzyme B, perforin, but this was just a very broad picture.
So this is in the tumor.
Yes, this is in the tumor. This is in the tumor. Yeah.
The lymphocytes in the tumor.
Yes.
So yeah, I guess like first the lymph nodes have to be viable, so the lymphocytes can get to the tumor based on the signal. So if you give a too much high dose, probably
You kill the immune cells.
Probably. Any other questions? Questions in Zoom? Is there any questions? Are they Jewish? I think they are not there. I think there's no questions.
Okay.
Questions? No questions?
Thank you.
Thank you. Thank you. Very good. It was very good talk. It was very interesting. Oops, I close the.
No, well, here we're stuck in the same problem. Like, I'm pretty sure you're thinking of the same thing as like. Photons, great. Energy efficient, double membrane, knowledge benefits, and I don't understand the link between that and it changes in well.
Yeah.
Like, there's a satellite, I don't know, it's three double membrane, but the cell die.
Yeah.
Well, they're saying, well, you're not actually getting the cell.
No, I didn't.